This is a much more of a production than the other one. It just feels like we're sitting.
But it's a very shitty.
The red light is on.
Production. Okay, hopefully I'm vaguely visible a little bit. This thing's very wide. Hopefully we learned something. Okay.
Yeah.
Hello and welcome to Journal Club.
Journal Pub.
Journal Pub. Oh. Okay.
I'm Lydia.
I'm Zach.
I'm Megan.
And I'm Bez.
And I'm Lydia.
Lydia, what is Journal Pub? This is Journal Pub, not club.
Journal Pub is where we drink and read a paper.
And we're hoping to do this in pubs in the future and learn the logistics of that. We don't quite have the logistics of that set up yet. We're going to read Unsupervised Restoration of a Complex Learned Behavior After Large-Scale Neuronal Perturbation. This is a paper, first author Bo Wang, last author Carlos Lois. We saw a talk by Carlos Lois. Me and Megan saw a talk by him at Stowers a few weeks ago that we thought was excellent. It's kind of on what this paper is and kind of on another paper, so we don't quite know this material. Should we keep trying? Yeah.
We can keep trying. I don't know. It's hard to edit, so we can't talk about meta things.
We could talk about meta things. Let's talk about it.
Well, my meta thing is you're talking to the camera. Are we talking with the camera? Are we pretending?
Oh, yeah.
I guess we're talking to each other.
Yeah.
Okay, fine. We're not talking to the camera. Goodbye. That's a good point.
That's true. This is Journal Pub.
It's a Journal Pub, yeah. Thank you for the
I think I
Moscow
will have trouble getting into it with talking to the camera.
Okay, what do you think of the Ithaca ginger beer? I love
this.
It's a little strong.
I really love this. I'm really like a lemonade person, and this is very lemonade.
But these are not our cups. They brought their own cups.
Oh, did you have 4 of these?
Oh, we have 4 of these.
I didn't see
okay.
that. I just started doing this first.
Yeah, we brought.
Those
we only own 2 cups.
Those ones went through a dishwasher, which apparently is not good for them.
These look nicer than our cups, to be honest.
No, yours are very like bright and shiny.
It's very Boston. I'm very happy about the
Did
Moscow
you
Mules.
request the Moscow Mules?
I did not. They
No.
offered.
Yeah, it's one of Zach's specialties.
This is like my go-to cocktail.
Right?
So
No, and the Ithaca ginger beer is the best. So we always whenever we go there, we bring a bunch back from us. So we have a lot.
Okay, I'm going to read the abstract. Try not to bore everyone too badly. Reliable execution of precise behaviors requires that brain circuits are resilient to variations in neuronal dynamics. Genetic perturbation of majority of excitatory neurons in HVC. What is HVC already?
That's a brain region.
A brain region involved in song production. Oh, yeah, I actually knew this. Okay, yeah, sorry. In adult songbirds with stereotypical songs triggered severe degradation of the song. So genetic perturbations triggered severe degradation of the song. The song fully recovered within 2 weeks, and substantial improvement occurred even when animals were prevented from singing during the recovery period, indicating that offline mechanisms...
I know.
What?
How do they prevent them from singing?
Indicating that offline mechanisms enable recovery in an unsupervised manner. Song restoration was accompanied by increased excitatory synaptic input to neighboring unmanipulated neurons in the same brain region. A model inspired by the behavioral and electrophysiological findings suggests that unsupervised single-cell and population-level homeostatic plasticity rules can support the functional restoration after large-scale disruption of networks that implement sequential dynamics. I don't know. It's a lot of... That's a big sentence. I'll try it again. A model inspired by the behavioral and electrophysiological findings suggests that unsupervised single-cell and population-level homeostatic plasticity rules can support the functional restoration after large-scale disruption of networks that implement sequential dynamics. No idea. These observations suggest the existence...
I think it's saying that it's happening on a physiological level, not on a learning level.
Right, that it kind of just if you perturb the brain region such that its song gets messed up, it can kind of spontaneously recover in 2 weeks without any sort of additional learning or external intervention. Like it just kind of figures it out.
Yeah. These observations suggest that
Finishing up, these observations suggest the existence of cellular and systems-level restorative mechanisms that ensure behavioral resilience. Okay.
Cool.
So, first paragraph on it.
Okay.
Why is this important?
Teeny tiny text.
Yeah.
Animal survival and reproduction require reliable execution of behaviors. True. However, neuronal representations change over time as a consequence of natural drift or due to neuronal perturbations caused by trauma, disease, or aging. What are the mechanisms that allow brains to maintain reliable behaviors over long periods of time, or after neuronal loss? To investigate this question, we studied the zebra finch, a songbird that, after learning a song as a juvenile, produces stereotypical renditions of the song with minimal variability over several years.
I like stereotypical here. To be like, I know that it means like, the stereotype, it's always the same song.
Yeah.
But here it's like, it's an unoriginal songbird.
Mm-hmm.
Songbirds have a series of brain nuclei dedicated for song learning and production called the song system. HVC, they're never going to define what that stands for.
I know, they're just like, it's HVC.
HVC is the promoter nucleus of the song system that projects to the motor nucleus RA, robust nucleus of the archopallium.
There we go.
Yeah, that clears things up. So that's something...
RA is robust nucleus of the archopallium.
So it's
Should
something
we
about...
drink every time we see the word HVC? Scouting it out. That seems like a yes. That seems like a yes.
I think that's a solid rule.
A solid start.
You're almost at the end.
Involved in song production, generating sparse sequential firing that controls the timing of the song. The circuit mechanism for the generation of this sequence is still under investigation. Am I supposed to popcorn?
Yeah.
Okay, Zach.
Previous experiments have shown that complete ablation of HVC
Drink.
Oh.
Complete ablation of HVC irreversibly abolishes song production. Similarly, ablation of nucleus UVA, a major source of input to HVC.
Drink. We might go through the drinks too quickly. It looks like it's just this paragraph, though.
Yeah.
Irreversibly blocks song production. In contrast, localized to partial lesions of HVC cause song degradation followed by recovery after varying amounts of time. Similarly, lesioning of nucleus NIF, one of the sources of input to HVC, also causes song degradation followed by recovery of neuronal dynamics
so this
and
is
behavior.
just like a really neuroscience introduction.
Yeah.
So I'm intimidated
I don't know.
at this point.
We might have to pause and try to figure out what the hell we're talking
We
about.
introduced a lot of new characters because there was like HVC and RA, which I was like down for.
Oh yeah.
And then
HVC
now-
is a big portion of the brain. I remember looking at a picture of that.
Or it's this little guy that's on the very top of the brain,
Yeah.
right there.
But I don't understand what the RA is because
They got a
the
picture.
nucleus
We're
is
skipping to
not-
figure 1 already. Figure 1A. By nucleus, they don't mean a cell.
Okay.
They mean like a center.
Okay, I hate this.
Yeah.
No, because I didn't want to interrupt the abstract portion of things, but they use plasticity in this stupid neuroscience way of meaning that it can change.
Plasticity is the opposite of plasticity. It's a science antonym.
Yeah.
Yeah.
So I just am so.
This is what we wanted to
backwards
do
on
after the paper draft that we never did. Is we wanted to do a science antonym draft of like what scientific words are like 2 words in 2 different fields that are
Wait,
like.
can you give me an example?
like plastic. Plastic in soft matter is like if you deform it, it stays deformed.
Oh,
Like,
which
that's
is like
plastic.
literally the
That's
opposite.
literally the opposite of
Okay.
plastic in neuroscience. So I just don't know where it came from.
Yeah. Oh, okay.
Okay.
So they got this
RA
HVC and
nucleus
RA.
is- yeah.
The RA
I think the
is involved
RA,
in the song production.
and it seems like that's a motor nucleus, so maybe it actually has to do with like signaling to things that actually cause it to, like be able to sing.
Okay.
And so if they get rid of the HVC, it doesn't create song. If they get rid of the nucleus UVA. Is that a different thing, or is that still
That's
the RNA?
a different thing, but like that's not even in the diagram, which leads me to believe that it's probably not that important.
Okay. If they get rid of little bits of HVC, they get song degradation followed by recovery. And I think that's what this is about.
Right.
Lesioning nucleus NIF, one of the sources of input to HVC, also creates degradation. Okay, so they're just like, we can mess with this system, we can get no song, or we can just get degradation.
And we kind of understand where HVC lies in this path.
Yeah.
Yeah.
All right, Megan, you're up.
Okay. The fact that we're there, right? Okay. The fact that the song is able to recover from these degraded states suggests the song circuit is quite resilient. Many potential mechanisms may contribute to this recovery, including homeostatic mechanisms, new page, and spike timing dependent plasticity within HVC, or even relearning of the song through the song learning circuitry.
I'm just going to say it, the word HVC shows up
I
in
forgot
almost every single sentence.
Yeah,
We do have more. We have all
Change the words.
Okay.
Okay. The potential cellular mechanisms within HVC that may underlie this resilience, however, have not been directly investigated.
She said HVC.
Oh my god.
Maybe the reader has to gets a pass.
That's true. That's true. So they can keep going. All right, reader gets the pass on HVC.
Nice.
I like the rule.
Okay. In this article, therefore, we use local and selective genetic manipulation of projection neurons within HVC and intracellular recording to probe the behavior dynamics and synaptic mechanisms of recovery after severe perturbation. To test hypotheses about how synaptic plasticity could account for the recovery, we developed a data-inspired model to explore the cellular mechanisms underlying the observed behavioral and electrophysiological findings.
I love the term data-inspired model.
It's not data, it's data-inspired.
I do like it.
I wasn't being
This is the thing I'm the
facetious. I liked it.
I'm most excited for about the paper so far because I like data-inspired models.
Okay. All right, results. So yeah, I'll stop when we get to figure 1A, and then we'll just basically go off the figures from then on. So I'll just read a few sentences.
Okay.
So the first part of results is song degradation and recovery after selective large-scale perturbation of projection neurons. Boo! Not a sentence. HVC, projection neurons, fire in an extremely sparse and precise manner when birds sing. To explore how singing may be affected by disrupting the precise firing of these neurons, we virally expressed a bacterial voltage-gated sodium channel, NaCH back. Okay, maybe sodium channel bacteria, in HVC projection neurons via a bilaterally injected lentiviral vector, LV figure 1A. Alright, so now we're just going to try to guess what's going on off of the figures.
And we'll go to the text when we need to, I think.
Which might be every single box. Okay.
But you guys have seen, like, he talked about this paper.
So, kind of. Like, all of these, like, bluey figures I recognize to be, like, spectrograms of the song that they recorded.
Oh, okay.
And presumably...
And then what are these UMAP projections, then?
Do you think those are UMAP projections on the right?
Yeah, it says UMAP 1
Well-
and UMAP 2.
No, I meant... Okay. Well, okay, let's...
I mean, like,
yeah.
this one, F, is clearly from B.
Let's go down the line. A is they're injecting this lentiviral virus into the HVC.
And it goes to the RA. No, the HVC signals to the RA.
Yeah.
Is
And
that-
so they're able to mess with individual neurons, I guess, is the claim.
So the expression... Sorry, I'm reading the results. The expression in neurons perturbs their activity due to two features. Do you want to know the features?
Sure.
Okay. First, NaCH back is activated at lower membrane potentials than vertebrate sodium channels. Okay. And second, while vertebrate sodium channel depolarizations last several milliseconds, those of NaCH back expressing neurons last up to greater than 1 second. Does that make sense?
This is biology.
I'm going back to the figure. Figure B, left. A spectrogram of a motif and the corresponding two-dimensional projection, middle, using the UMAP algorithm from an unperturbed animal. Oh. So this motif, he said, was like a bit of a song, right? So this is a bit of a song. The UMAP is not a genetics UMAP. It's a song UMAP.
So then are they just taking the raw spectrograms, doing dimensionality reduction, just putting in a UMAP, and then are they getting the syllables from the UMAP, or did they like they already knew what a syllable was?
I think they've defined that.
I think that they've temporally defined a syllable, right? Because they go I, 1, 2, 1, 2, 1, 2. So there's syllables in a motif, right? A syllable is like a letter or something in the song.
So the syllables are labeled on the bottom of B, right?
And then they make the UMAP, and they show where the syllables fall on the UMAP. But I think that they manually allocate that.
That's like already a thing, probably, that they knew about.
Okay. C, firing pattern of retinoic acid not retinoic acid. It's this RA portion of the brain. Firing pattern of RA projecting HVC neurons with NACH back plus or without NACH back control.
I think the takeaway is that NACH back like messes with things.
Yeah, it slows down the recovery.
Is that what it's showing?
Yeah, so I guess the potential shoots up, it comes right back down with the control, with the NACH back, the potential shoots up.
Okay, and it never goes down and then is able to shoot back up.
But what does it mean, potential? I guess this is how they actually make the measurement. Did they have a little probe and it's just an electronics probe?
Yeah, they didn't talk about
I always thought that these probes had like some frequency that they were reading, that the like cells were firing hundreds of times per second or something like this. But that's not what this looks like, right?
Oh, so it does say it's fused to a green fluorescent protein, allowing for visual identification of the infected neurons. Extended data. Do we have supplemental figures?
No, I didn't do them.
Oh, okay.
If they put it in supplemental, we cannot learn about it. Right? That's the rule.
Okay, yeah, so they did whole cell patch clamp recording.
Methods.
Yeah, I can put the methods in. So D is example spectrograms and the UMAP visualizations, which I think are just art. I don't think the UMAPs tell you really anything.
Well, I mean,
you can tell that
it kind of tells
the
like
second and third are more similar.
It tells you something,
Sure.
right? Like
okay, so there's a perturbation.
F1 and F.
Yeah, you're right.
4 and
You're
5
right.
are very similar to.
Yeah, so you've got a perturbation, then you have it collapses down into this very trivial UMAP because they've forgotten most of the syllables, and then the perturbation or the thing recovers. Yeah, the UMAP recovers.
Wait,
so I don't like what's going on.
Yeah, so they're showing for D, like following the. It's hard to. Yeah, you can see it in the spectrograms. You can also watch the UMAPs recover their shape.
Oh,
which I guess is nifty.
Well, I don't understand which way
Time's
going.
going down.
So
6
DPI days post. Yeah.
Okay. So this is initial.
Oh, so this
Well, so
is
this
just
is a
like
different
they
figure, so
let
that's
it
like D
sing.
is a figure, and then
Yeah,
on
but like
different days.
this is initial
Yeah.
for each of them,
After,
and then
Yeah.
the injection.
Oh, that's cool.
okay, yeah, I feel the UMAPs are useful for visual representation.
I'm sold on it.
What's the difference between D and F?
Yeah, that's my question.
So F is example spectrograms and corresponding UMAP visualizations of song renditions of a bird at different DPI with LV tent.
Ooh.
LV tent is going to be a new thing.
Yeah, I think we haven't introduced LV10.
So we will have to read what is LV tent. 1D.
NACH back. To investigate whether directly silencing HVC projection neurons is sufficient to degrade the song, and if so, whether the song can recover, we delivered the same type of lentivirus, but carrying the light chain of tetanus toxin. Tent is an enzyme that cleaves synaptobrevin, a protein essential for the release of neurotransmitters. Thus, expression of tent in a neuron does not alter its electrical activity, but abolishes its ability to communicate with its postsynaptic targets. Paired whole cell patch clamp recordings of brain slices showed that expression of tent in HVC RA neurons permanently abolishes their excitatory, nobody drank, their onto inhibitory interneurons in HVC. Let's see if you were on time with the first one. Furthermore, the excitatory drive from HVC received by projection neurons in the downstream nucleus, RA, is substantially reduced.
We do have to be a little careful about going back to the main text for information, because we're going to have to drink a lot.
That's true.
Well, we could
don't
change
say
it.
it.
Only when we read it. I don't think we have to do it every time we say it, right?
No, not every time we say it, yeah.
Okay, so it's another way of messing up the HVC. Only this time it can still fire, it's just it can't project to its downstream neurons.
Right.
Was that accurate?
I think that's what it was saying. It abolishes its ability to communicate with its postsynaptic targets.
Wait, so why do these UMAP look different? It's just each bird gets its own UMAP?
That's my, I'm not
I think
totally
each
sure. I thought they
song
were supposed to have stereotypical.
Gets its own UMAP, and the song in D is clearly not the same song in F, even before the perturbation.
Wait, because even in B, these are two different UMAPs, right?
No, I think that this is a grayscale of this.
okay.
Yeah, I think that those are literally the same.
But like F is clearly the same as B.
Yes, okay. Okay, and then maybe D is like, this is what this bird's UMAP looks like?
I don't know whether it's this particular bird or whether it's a slightly different song for some reason, but I thought that
It does kind of look different.
these animals, which ones are these? These are zebra finch.
Mm-hmm.
I thought they only had the one mating song.
I think they do.
And
And I
they're all
think it's
supposed
all-
to be the same, but maybe there's still bird-to-bird variability.
I mean, they got hundreds of songs, right? They don't all sing the same song all the time, right?
Are you sure?
I thought the whole point was that it was stereotyped.
I think stereotyped among many songs. There's no way that zebra finches only have one song.
The talk that I went to was about like the zebra finch mating song, which is passed down.
Which is
from-
always the same song.
Okay, but then maybe
Father,
there's
son.
a zebra finch. Because do you remember there was this lady that talked about like there's a
Well,
song
wait,
for
wait,
let's
wait.
all go together and like go somewhere.
Wait,
So I think there are different songs for
Different
different
songs,
things.
the spectrograms that we get here, those are neuronal spectrograms, right? Not
birds,
audio.
Are these birds already?
Oh, okay.
Okay, so it's not that the
Oh,
Birds
it's
are
not
already
the
messed up, the
song.
songs
It's the neuron. It's like the.
This isn't an
antigen.
audio.
This isn't an audio, right? This is it. This is literally the neuron firing, right?
Is this
Am I
free
wrong?
I thought
Oh,
it was. I thought it was
it's like-
audio.
It has hertz. I guess that'd still be speed of firing.
Or do well,
I thought it was audio.
or it's the because they said they do like patch clamp.
It is this 10,000 hertz makes me think that it's audio. That I have difficulty seeing how a cell could fire that fast.
I think this is audio.
Okay, so
Yeah,
these are
like
audio.
he
Yeah.
didn't play anything during his talk.
There
Did they
was
play any
there were
songs?
there were songs. There were songs. I don't remember there being like
I
totally
just thought the
different songs.
spectrograms were neuron firings in his talk. If this is what is the heat map scale
It is the-
amplitude?
So seeing where we're coming from, that we're so spectrogram of motif.
I think that
Are
must
they-
be
are they-
sound.
We can
Elfin
note the method.
10 birds already messed up.
Oh, actually, I'm even more confused.
Yeah.
It says that this spectrogram, this UMAP that we're looking at, is a UMAP containing 100 songs of the birds, randomly sampled one day without perturbation.
Yeah, so continuous audio recordings were segmented into individual beds.
Yeah, sure.
So it's all
Okay, so it's
audio.
all audio. This UMAP is the average of 100 songs of the bird. But it's the same UMAP as in F.
Well, because that's that bird's UMAP, I think. And then he got perturbed.
Oh, that's just
It's a single.
Oh, it's the
versus-
one bird. Yeah.
That's his normal song, and then this is
Right.
perturbed in the recovery.
Yeah.
And this is one bird and this is a different bird,
Yeah.
right?
Yeah.
I mean, it has to be. You can't.
But why is
And then
their
he
song?
got perturbed.
Yeah, why is their
Why is
song
your
so
song so
different?
different?
Well, because I assume the UMAP is made from just this like continuous recording for the whole day.
Yeah, I guess the question is Yeah, the two birds. They couldn't get two birds that sing the same song.
Well, but maybe the UMAP is not very good at being like capturing the similarities between birds, right?
Pretty different.
I always forget this about this. I think UMAPs are stochastic, right? That like you can run a UMAP on the same data set twice and you'll get two different shapes.
I think so, right?
Without a seed, I think you will get it.
Yeah, so they could be literally the same UMAP. It's just hard to produce the same UMAP from two different data sets, even if the data sets are almost or are identical.
Well,
I
I
guess
was wondering, are the birds already kind of messed up? Like
I think the initial bird. Well, I mean, they
Yeah,
got holes in
ten.
their brains, but you know.
I think the first line is supposed to be before the
Yeah.
injection, and then they're.
I mean, it's a good question. It says, it says pre-perturbation. Yeah.
Yeah, and then it says I is introductory note and C is call
Introductory note
on here.
and call.
so is the introductory note the bird, or is that to get the bird to sing?
I think it must be the bird.
They must have like
That must be the first two
They
motifs
must
that
have a
it
whole,
seems
like-
are like little.
description of they probably have a bunch of ways they describe the songs, and that's part of the bird song.
Okay, figure 2. Dynamics of song degradation and recovery after large-scale perturbation of HVC projection neurons. Again, not a sentence.
I think we're going across the table. Oh, we're not reading.
We're not reading, we're
Okay.
just doing
We're
figures.
just.
Is that a sentence?
I mean, it doesn't have to be a sentence to be a figure title, right?
We were taught in physics that it has to be a sentence to be a figure title, and people would throw things at us if it wasn't.
It ends in a period.
It does end in a period, and now I'm like, it kind of looks like it has, like,
No, it
nouns.
doesn't have a verb.
Is degradation and recovery not a verb?
No, those are both nouns in this context. Song degradation and recovery.
Okay, it doesn't have a verb. A, syllable duration in milliseconds. We've moved off of, and then DPI. So this
Days
is-
post-infection.
Yeah. And this is LV tent, and so these are two of the lentiviruses that mess with the bird. You mess with the bird, and then it recovers from figure A. Seems clear enough.
Yeah. It's like a heat map and it's like all messed up and then it like resolves.
And then they have a kind of one of these. Look at this. A part B, the average syllable duration. This is a terrible graph. I hate this graph.
Control, degraded, recovered. Average
So this-
syllable.
Each line
Individuals.
represents one animal. Yes, it says each line represents one animal. That actually makes me hate it a little bit less.
Okay, so it's a way to show multiple animals, like the
Yeah. All right, so then B, density distribution of the maximal log power of syllables day by day. Again, it's just A, B, and C are just three ways of saying our lentiviruses.
Okay, but are we supposed to see differences between the tent and the NHC back?
It looks like the tent is like
Or they're supposed to be the same?
More
Is that what they're claiming?
disruptive.
Disruptive for longer.
But either way, it recovers.
It
And I think
recovers.
that must be the point.
The point.
That there's, again, the difference between tent and NACH back, well, obvious to them, I think is subtle to me.
Okay,
Mm-hmm.
so we'll just think of them as the same thing for now.
And I think they're going to make a point that they're different, or like that because they do the same thing, you like learn something about their brain or something like this. Figure D is a
Wait,
schematic.
Can we talk about what is mean entropy?
Ooh, there's a word, mean entropy. Great.
This one.
Syllable mean entropy.
Like, how would you define
The
that?
averaged entropy of all syllables at different time points. To the methods.
So see like how different the syllable is.
Oh, because it
over
does.
each of
It
the songs.
does. It increases when the neurons are like
I
messed up.
really thought there was going to be like an entropy equation in the methods, and there's not.
In nature neuroscience, you thought there would be an entropy equation?
I did.
I think it's probably like the variance of syllable duration.
There are some equations back here.
Yeah. But none of them are entropy. Oh, I'm quite sad actually. Well, so then actually I don't know what they mean by entropy. Do they just mean variance? I guess
so, right?
2C.
Duration distributions of best syllables. Command F entropy.
Yeah, that is a problem with having it fully paper. Yeah, I don't know.
Okay, it says syllable feature distributions. Syllable features include duration, log power, and entropy.
Is there like an obvious definition of entropy in this case?
I have no idea.
I assume it's how, when they say the same thing, how different it is. Like it means that it's very,
So like variance, kind of.
Yeah, but like, why not
Why
use the
say
word variance?
Entropy and not variance. It just sounds like it's something that maybe is used in the field.
Yeah.
Because it says syllable features, duration, log power, and entropy.
Yeah, no idea what it means.
So,
Alright,
well,
big
I'm
X on
sorry.
that part of the graph. D. A schematic showing methods for tracking the change in song over time using acoustic features sampled from the spectrograms. So they use mean frequency, pitch, entropy, and goodness of pitch. I like that. Which is just variance on the pitch, right? Then it looks like they score the features somehow. They break it down into a single number. And then they do principal component analysis again. And why are we doing principal component analysis?
I mean, I'm hoping that it's going
Oh.
DPIs.
Yeah. So originally, the original syllable overlaps on itself, duh.
Mm-hmm.
Then the infection moves the two away, and then they come back to each other.
But first, they go further away.
Yeah, because the infection is taking place, right?
Okay.
Well, yeah, like how long does this infection...
Wait, so where is it the furthest? Like 5 and 6?
I would say 3 and 5 are the furthest.
Oh, yeah.
I guess there's a question of
Oh,
how
yeah,
long does
most
it
of
take
it,
the lenti... Because once the lentivirus gets into the cell is doomed, right? And it's just like a question of how long...
So you think that's just a length of you don't think it's a...
Well, essentially that's my question, is like, is the cell still a little bit alive one day
I think
after
it's...
infection?
Well, I don't think the cell dies.
I don't think the cell dies. I thought it was just that it wasn't
It's
doing
like
its
not
job
firing.
quite as
Yeah.
well.
Well, I guess two things here. One is like, is the cell still doing its job a little bit one day after infection? Two, like how quickly is the cell? The other is lentivirus into a cell. I feel like the cell is eventually going to die. Like one thing we've learned about using lentiviruses is they just keep producing and producing and producing and producing until the cell is doing nothing but making the virus. That seems to be our lesson from using lentiviruses.
And so then what's happening here is it's not... Yeah, like over time, the lentivirus is getting diluted out.
I mean, the lentiviruses are competent, right? So they make more virus. Right?
I mean,
no,
like, for the-
lentiviruses, it won't make more of itself. It's not replication competent.
RCAS is replication.
Yeah,
Lentivirus
so
is not.
it'll get in.
Okay,
It might
okay.
even get silenced.
Okay. So the cell actually will win this, or can win this fight.
Yeah.
It just has to survive long enough. Okay. So why show this? I mean, we were pretty convinced by like A. Why are they doing this?
I guess maybe they're trying to convince us that it recovers to exactly how it was before. Because maybe we could be like, oh, it's slightly different than it was before.
Yeah, I believe that. I believe that their data, it looks like it comes back. F and G look less convincing, and I don't want to think about them, but you know. It looks like, yes, the song degrades and recovers after large-scale perturbations.
Is that all this is showing? Is just...
Well, the whole figure says that it's showing the dynamics of the song degradation and recovery after large-scale perturbations. And this is all about the song itself.
You think that this most degraded time scale is set by the lentivirus infection time scale?
I don't.
So then
I think the lentivirus... Well...
What is causing it to get more degraded before it starts healing itself?
I think this is how long it sticks around.
The lentivirus?
Yeah.
So it's...
Yeah, or it's possible that maybe it takes a couple of days before it's like really expressing its cargo and like messing up the cell. Or maybe like it's expressing and then it's like clogging up the system.
But I think
the
time
delay
scale
is due
here
to lentivirus, not due to some like...
Well, I could
Like
see
you have
it.
to mess yourself up to get better sort of network problem.
Oh, that's fair.
It's an interesting idea. I think we can't like possibly know without like put a single cell in a dish and like watch the dynamics of the lentivirus itself.
Okay.
I think they can't possibly
Well, but
know
like,
either.
okay, so then they don't know why they're showing this?
No, I mean they're showing that it goes, it leaves, and it comes back. Do you think that they're interested in the time scale?
It
Well,
gets
like,
worse for
it's
4
a-
days and then it comes back.
The dynamics of song degradation and recovery after large-scale perturbation. So if they only cared about the end state, then you wouldn't care about the dynamics.
If they cared about the dynamics in the way that you care about the dynamics, then F would have an averaged line and there would be a fit.
But it could totally be something about the biology. Or it could even be like the half-life of the protein for the lentivirus where it's like inhibiting the neuron from firing and then maybe it degrades and then it's like
Yeah,
fine.
and this is when its peak,
Yeah, I
like
think its
the
peak is
peak
at
concentration is going to
day
be at 3
3.
days.
Well, that's what, they state that, right, actually.
Okay.
Okay, figure 3.
So, I guess you're saying it takes 24 days to get back?
Yeah.
17 on the left.
17.
Wait. Oh no, I guess 24. You're right. 27.
Yeah.
Okay, it's all we're reading. It's all one. Why are they showing us so many time points? I really think we're missing something here.
I think it's just that there's a little bit of a style here of tiny subpanels in big figures, right?
Well, I think maybe
But like,
they're
what
really...
would have been missed by just
1
Just
DP or negative 3
one-
DPI, 1 DPI, 27 DPI.
Yeah.
Why not just have 3?
unless you're trying to highlight this
These
upturn,
literal
downturn.
like plume dynamics.
Yeah.
I mean, I guess the cool thing about this field is they kind of just have to record the birds all day for 30 days, and then they have like all this data.
Yeah.
You know what I mean?
And
Like,
so they might as well put it all in the figure.
Well, okay.
yeah.
If you're gonna poke at it so much, let's just check the text real quick. What do they say about 2E? We measured the acoustic distortion of songs by calculating the distance of disordered syllables from the original syllables before perturbation, normalized to the maxima in a high-dimensional acoustic space derived from the spectrograms. That's all they say. So yeah, they just thought it looked nice.
It does look nice.
Figure 3. Substantial song recovery without practice. Alright, so this is like one of the main claims by the abstract is that how do they song prevent? That's what I want to know.
Oh, I don't know if I'm gonna like the answer.
No, they can sing later, so it can't be. If they just like keep them in the dark or something, or they not?
Yeah.
They were fitted with custom-made fabric vests and a 20 to 40 gram weight attached to their vest, pulling them toward the ground to prevent them from acquiring singing posture.
It's okay.
That's so sad.
I don't know. I'm that's less sad than I thought it was gonna be.
Yeah. I thought they were gonna
A
like
vest?
muzzle them.
Oh, okay, so
That
it's
doesn't
like...
seem that
They can't-
different
they
than like
can't sit up,
Oh,
so I guess they can't.
that doesn't
sing
seem
about
that
it.
different from like hooding a falcon or something.
okay. It could have been worse.
It could have been worse is all I'm saying, yeah. Again, these birds have holes in their brains, so or holes in their skulls. Okay, so pre-perturbation, it's got a song. C, the last 10 bouts before prevention. So again, a bout is like a motif, is like a syllable, and the bout is like a part of the song from his talk. The first 10 bouts after prevention. And so the point is the last 10 bouts. Okay, so C is they've been perturbed. They haven't been prevented from practicing yet, but they've lost their song. So C, you're supposed to see they've lost their song. And then D, you're supposed to see they've regained their song 13 days later, even though
They haven't
sung.
seen.
I don't know. They look like they're doing pretty well in C. Don't you think?
I think C, the point is that compared to B,
No, compared to Peer.
yeah,
Oh,
B
okay,
is the
okay.
song. And so the point is that they've got this 1, 2, 3, 4, 5, 6 in D
Okay.
that shows this song, and they show the same like pre-perturbation 1, 2, 3, 4, 5, 6, 7. And this
So are they by themselves, or are they like hearing other birds
that's
singing?
a great question.
Right by themselves.
You think they're,
You think they're by themselves?
I don't know.
The like logistics of that aviary, where like every zebra finch gets its soundproof box.
Or
Yeah.
yeah, maybe they
I think
go.
they must. How
No
else do you get
way.
these song things?
No way.
No, like you're recording all the birds all
How
at
do you-
once.
how do you
Like how
tell
do you
apart?
disentangle the different songs?
Sure, each bird that you're recording for the recording session gets its own box.
Yeah,
But
and
this
for
bird
the-
is not going to get its own soundproof box for 13 days.
Why not?
Oh, okay, so they were able to
He can't
move.
fly.
He's got a jacket on.
They were able to move, perch,
Probably in a
drink, and
shoebox.
eat friendly, and even allowed to sing several renditions occasionally.
Well, they're wait,
Well,
what?
They had
Wait,
they had the jacket on.
wait, they were allowed to sing sometimes.
Well, just like
Maybe they
for their
can.
own.
Maybe they can. They just.
It says even allowed and we confirmed and even allowed them to sing several renditions occasionally. Okay, so it doesn't like totally like it vastly decreases.
It vastly
the-
decreases. So
yeah.
it's saying that practicing is not really the mechanism for recovery.
Adult male birds were single housed in sound isolation cages with a 14 10-hour light dark cycle for greater than 5 days until they were habituated to the new environment and started singing. Thereafter, birds were kept in isolation until the end of the experiment.
There you go.
God, that's just a lot of space.
But they're small. I mean, like they're small.
If we start single housing mice, the mouse house fills up instantly, and also the
That's just like
Yeah, but they
lines.
only have like 5 individuals, really.
okay.
Yeah.
Okay.
They
I'm
probably
thinking
just
about
have, like,
hundreds
10
of birds. Yeah, interesting. They've only got like 5, 10 birds. Interesting. Okay.
Yeah, because I was curious though if you perturbed it and then exposed it to like new songs, if it was like plastic enough to be like, oh, that's the song. But it seems like it's pretty like locked in to like the song that they already knew.
The rest of this figure, E, F, G, H, seems like they're just trying to convince you that it had substantial song recovery.
I just am convinced that they have song recovery. Like, is this what?
But they had song recovery even though they weren't allowed to practice.
Oh, okay.
Yeah. Could have been an email, no. Figure 4. Songs continue to recover rapidly during the post-prevention period. Yes,
Post prevention.
so you do song prevention.
Oh,
So the
okay.
question is, yeah, can they get back to
To where they were before.
Yeah, so
like,
they,
but can they do it faster? So is the recovery faster if they are prevented?
Mhm.
Which I don't know what their claim is. If it is or it is not.
each color represents one bird.
Relation.
Maximal
Freezing.
degradation by
That is
13
something.
days post-infection. The free singing has less maximal degradation than the prevented singing. So they lose more of their song if they're prevented from singing.
Okay.
But that's at 13 days.
At 13 days, yeah.
Yeah.
And then at what's the D?
D is scale distance to original syllable. It's how close back to the original song did they get. And it does seem, red seems to get better. So which one's red?
Is that the prevented?
It's with, no, black is, with is black. So with song prevention is black. So they
It took them 128 practices
yeah,
to
it
get
takes,
back to half as good as a song.
with song prevention.
Or half.
Without, yeah. So if they don't do the song prevention, the birds get it more correctly and they get it more correctly faster.
Oh, okay.
So they can get it back with song prevention, but song prevention really does make it harder for them to get it
Limits,
back.
Yeah.
limits there.
Okay.
But it's not the time, it's the number of practices.
Practice epochs. Yeah. But
I mean, they don't
I'm confused about
Quite sure that.
Well, this.
I feel like that there's probably no way to disentangle practice epochs and time.
Well, with song
how many times
prevention,
they try, right?
song prevention, there's a way to.
So
But
the
then it
black
does-
is prevented. The red is normal.
But then if you look, I'm confused about comparing D to E,
So E is practice epochs to half recovery.
which I would assume would be this P1 half.
That's, the half is like how long
Yeah.
it takes to
Are we
Yeah.
switching black and
Yeah,
red?
yeah, this is what I'm confused about.
I think we're
D says
they're
example.
mislabeled.
Yeah. So I'm going to read D again. Example plots of syllable recovery versus the number of practice epochs with
I just
song
think
prevention
I
is
think it's mislabeled.
black trace or without song prevention red
Right.
trace.
That makes sense. It takes them 22 practices to
But
get
it's just
back.
the
But if
opposite
you
of
look
E.
at E,
Oh, I
E
haven't
is
looked at
the
it. I haven't
practice
looked at
epochs to half recovery. So it's like taking all of the data, all of the birds that they measured for free singing are the black ones, and it takes them way longer, way more practice epochs than the prevented singing ones.
What is blue, red? Oh, these are the individuals. Why
I'm going to the text to see what they say about it.
in the free singing?
Yeah, see.
So
Lenny has a good point.
In the prevented,
Yeah.
they know the individuals, and then in the free singing, presumably they.
Also know the individuals?
Well,
or
but
no?
why is it black versus the 3 colors?
But that's what I'm saying. I think they're switching up the colors.
I think they're definitely switching the colors between the 2 figures, but I'm confused by why
Why
the
they're
free
all
singing
black.
the free singing ones are all black.
I think they still must know the individuals because I think column is also.
Oh, column is individual.
So it's just
Okay.
yeah.
But what is the difference between?
We measured the full trajectory of song recovery as a function of practice instead of time, and observed that birds that were prevented from singing made substantial recovery with far less practice than birds who were
Yeah.
allowed to sing freely.
That's so they just mislabeled D. That's unfortunate.
Okay.
And they mislabeled D because
Right.
the text is so tricky. The with
Oh, so preventing
without.
singing actually improves recovery.
Well, improves recovery if you're measuring by metric of number of practice
Yeah.
times.
oh,
so, like, preventing
because it was
singing-
just time. It wasn't. It wasn't the practice they needed. It was the time.
Yeah,
Okay.
yeah. But the practice doesn't
Ah.
actually help them
Recover faster. Oh, I see what you're saying. Because the free singing, they can practice that whole time.
And so they practice a ton,
yeah.
but
But it doesn't
takes-
actually
it doesn't
help.
help them at all. And so we just
Ah.
shift this over.
But doesn't it help them?
It doesn't help them. I mean, it.
You took them 22 versus 128.
No, 100.
practices.
So this is what I'm saying is that 128 is the free singing.
Yeah.
Mm-hmm.
It's the free singing. Like this
Oh.
is
Yeah, it's just
That
wrong in
E
the figure
is-
caption.
E is the average of.
Oh my gosh, this is so exciting.
All
We
of
found
the-
a textual error
Gross.
in our first journal pub.
Gross. We're gonna find a textual error every time. It's because they didn't put it in the figure itself, right? Which is what they should have done. They should have labeled the graph and the thing, and then they have this really tricky wording with black trace or without red trace song prevention that they just during some edit flipped them by accident.
Yeah.
But without song,
Yeah, they're just wrong, right?
without song prevention,
Look at E.
it goes back faster.
E is right.
So this is without sign.
Yeah.
So they
No, it's not.
yes it is. The legend is wrong.
If you go to the text.
Because they're saying the free singing birds got to practice a lot more, but they didn't recover faster with more practices. Whereas the prevented singing birds, they didn't get to practice that much, but when they did get to practice, they recovered really quickly.
And they recovered better, right? Because the red is better.
Yeah, well, but it's also just they just didn't sing for 2 weeks and then.
No, but I mean
Okay, so
the
Oh, okay.
distance to original syllable is smaller. This is actually a really
Where's
unfortunate
distance?
typo. So D, the y-axis, is scale distance to original syllable. The red bird that got to practice less gets closer to the original song.
What are you talking about? They're about
You
the
mean the
same.
height
back
of the
to one.
thing?
One. Oh, practice epochs post-induction. Okay, sorry. I misunderstood. You're right. They both get back to 1.
Yeah.
It's fine. Yeah. Okay, yeah. I'm sorry.
I mean, that would be fascinating. But I feel like this is, it's interesting.
Yeah.
Like, yeah.
I
remember-
guess it kind of suggests that like learning and practice doesn't actually matter. It's just that
You don't have to physically practice. You can
Think about it.
think about practicing, which jives with my experience practicing piano.
you
I
have
don't
to think
know.
about practicing.
That's what I did in high
They're
school.
not learning necessarily a new skill. They had brain damage and they're recovering a skill that they had.
So they're not thinking
they're-
about
yeah,
practicing.
they're-
So if I
They're
get
just-
hit by a football player, I don't need to practice talking to regain my speech after that.
Well, sometimes you do.
Yeah, like there
You
could
have
be
to
a
go to
scenario
speech
in which
therapy.
you.
They're
Yeah.
suggesting that it doesn't matter if I do or not.
Well,
have to
for
wait.
this particular
Yeah,
perturbation.
Yeah, and
They're
assuming
saying
that I
that
have
this-
this HVC organ.
I mean, I think
in
this
my
is a
brain, which I
really
don't.
weird. Yeah, like my understanding of this system is that it's pretty weird.
Yeah. It's definitely a very unique,
Yeah.
brain thing that we don't have. All right, figure 5. Population level homeostatic plasticity and recruitment of silent neurons in a network model contribute to the recovery of sequential activity. When I read something out loud, I can't hear myself say, and then I have no
I
idea what
mean,
the hell I just said.
Like,
Fairhall
I didn't
Carlos
understand what
Lois.
you said.
Population level homeostatic plasticity and a recruitment of silent neurons in a network model contribute to the recovery of sequential activity.
Okay, so this is their modeling figure.
Is that true?
Yeah,
Maybe
network
it's in
model.
the- Yeah.
I would have put that earlier in the figure title, but
Population level homeostatic plasticity, no idea what that could possibly mean. And recruitment of silent neurons in a network model
also no idea what that means.
contribute to the recovery of sequential activity.
Okay, so they did whole cell recordings in the HVC neurons in birds injected with the LV tent or controls.
GFP control. Yeah.
Or G control. What does that mean?
What's a whole cell recording?
I guess, is that the patch clamp?
That's the patch clamp. Okay.
Okay, I have to go.
But this isn't
To
not
the
then?
text.
I'm going to read the beginning of the section, this has section 5, out loud, because it starts with something that looks like actual text. There will be some drinking involved. What are the cellular mechanisms by which the brain restores the precise execution of the song after such drastic perturbations? Dual whole cell recordings between HVC-RA neurons and interneurons indicated that expression of TNT leads to permanent muting of the infected cells. As both LV manipulations, lentivirus manipulations, permanently perturbed the infected neurons, both LV manipulations being the NACH back and tent manipulations, permanently perturbed the infected neurons, reducing the number of functioning HVC projected neurons. We hypothesized that the recovery may depend on changes among other neurons that were not genetically perturbed, unmanipulated neurons within the same HVC. I said HVC twice.
I think
I'm gonna get a drink. Zach didn't drink. Megan drank a little bit of Zach's.
We ran out.
Oh, you ran out. Oh, should we pause? Should we get more drinks?
Do you need more? How are
I'm
you guys
pretty
doing?
empty. I could finish right now.
Do you want another? Do we have enough stuff for
We have enough.
another round?
Okay.
Probably.
All right. Let's
it
pause.
might be light on the ginger beer, but we'll make it work.
This is a good pause point, figure 5.
I think we have to go back.
Did you get to go back?
Well, I was reading while we were talking silently to myself because sometimes reading out loud is hard. But the conclusion from figure 2, I don't think we fully.
I
We get it
still
wrong.
think these are completely fine.
No!
Do you-
No. Wrong.
Okay. Pause.
How is it different?
Because this isn't time. This is
The
number of practices.
Yeah.
So the free singing bird had a lot of practice. right?
Yes,
Over
it took
its
him
like
more
time.
more practices to get back to normal.
Yeah. Whereas this guy, because he wasn't allowed to sing, he didn't get a lot of practice.
So we didn't lose
when he did
the
start practicing.
phone's recording.
The song recovered very quickly.
When he did start practicing.
Yes.
Yes, that makes
So
what's
this,
wrong
so
with-
yeah.
How's that not consistent with this?
Okay, wait. Go.
So the
Wait. This is for because we're still recording now. I mean, we are still recording now, but like, let's have drinks and then we'll pick it up again.
Okay, the only reason we said it was wrong is because in the legend it says the number of practice epochs with black trace or without red trace. So it's saying that red trace is without song prevention, and it's saying that black trace is with song prevention, which is wrong.
Wrong!
No, this is without song prevention, right?
Okay, that's black.
No, and then it says with.
Black trace.
No, with
With is without. I mean, black is without.
with song prevention is black. Without song prevention is red.
But why? Like, how do you? I don't understand. What is?
So the legend.
It's making me afraid that I'm wrong.
The
right? Like,
legend says that
I'm
the
so
legend says
convinced that I'm right,
that
that
song
I
prevention
can
is
be
black,
Okay.
and song prevention is clearly red.
No, I think that's what they're saying is that the song prevention, they weren't allowed to sing, and then when they are allowed to sing, they can get it right faster.
Yes, but that's
Yeah.
How is that not the same? Like,
Because with practice epochs with song prevention, right? With song prevention.
yeah, they can't sing, so.
Would be red.
But
Why? What do you? How can you? I mean, they get to pick the colors. I don't understand.
No,
they
because-
say
Where are you saying it's different than in the text or in?
No, I'm saying this is different than this.
But here doesn't make a claim.
Yeah, it says
with song prevention is black is what the text says. With song prevention is black in the text.
in the figure caption.
and without
Which figure can?
right here.
D.
Yeah, I'm reading that.
With
Black trace song prevention.
Yes, with song prevention. Okay.
But
but
clearly
wait,
the
this
black
is,
trace is not with
Is
song
this
prevention.
with or without song prevention?
But how can you know by looking at the data which is right?
Because this matches that.
It does. This black and this black work together.
Yes,
Yeah.
I agree.
Yeah.
I agree.
Yeah. So these
Yeah.
are consistent.
Yeah.
Yes.
These are consistent.
Yes,
Yes.
I agree.
But
the legend
The
is
legend is
for
wrong.
singing. So this legend, either the legend or these, one of the. Okay, I see what you're
Right.
saying.
Well, something has to be wrong, and then the main text.
Wait, I
makes it
wasn't
seem like-
looking at.
At the
wasn't
legend.
looking
I
at
see.
this
So you were saying that this was
I
fine.
was saying that
Yeah,
if this
yeah.
is
Yeah,
correct,
I
then this
agree.
one's wrong.
Yeah,
You guys
yeah.
are saying. Okay.
No, but yeah, in the legend it was just saying that this is
Okay.
black and this is red.
Yes. Okay, I get it now. Yeah, one of them is wrong, but we actually can't tell because we didn't read.
But we did read,
Well,
and
if
so we
you-
did find out what
Yeah,
was
yeah.
actually wrong.
I got it.
Yeah,
Yeah, I was trying to figure out how you could say which one of these, like, these aren't labeled.
Right.
So
right,
I was
yeah,
like,
yeah.
you're trying to say that you know which one is which.
Yeah. And this is, I think, why the error came about is because they didn't label it in the figure. I didn't print that. If I printed this with the extended data, it was like 35 pages per copy.
Yeah, no, that's too much.
All right, figure 5. We've got A, which shows a schematic illustrating whole cell recordings performed in HVC neurons in birds injected with the LV tent or naive controls. And then B is example traces of MEPSC recordings, which I'm assuming is patch clamp. C is group data of MPSC's degraded indicates recordings at 5 dpi, recovers indicates recordings at 25 to 35 dpi. So, normalized MEPSC frequency, so patch clamp frequency. For
So
the
is
control,
this they just put a probe on the cell
yeah,
and then they measure
they measure how often it's firing.
firing?
Yeah.
Okay.
And I think like 100 hertz is like a normal, but I guess this isn't in hertz, this is percentage.
Well, so they say, and they have the control. there.
Yeah, so the control is 100 percentage by, standard. The frequency looks like it doubles in the degraded, and What is this GFP minus, GFP plus? What's the difference between GFP minus and GFP plus?
Is it
With and without the...
the lentivirus? Right?
Yes. So GFP minus is just GFP. No, GFP minus is just the...
Is nothing.
What is GFP minus?
I think I think they're patch clamping a cell that didn't get the virus, but that's in the HVC.
And it got injected.
But they have a naive control in A.
But don't they want to...
Lentivirus with tent or the pink doesn't have the tent.
No, but naive controls, no virus, is gray.
Because that
I
bird
agree.
didn't get injected.
Yeah.
So what is the difference between GFP minus and
So
GFP
like
plus?
the injection might affect their singing. The fact of
But I think the
being
naive
stabbed.
control did get injected. There was just no virus. Like they have a little injection thing in A.
Well, that's the patch clamp thing, no?
Oh, is that the patch clamp? I thought that was the injection.
I think that's the little patch clampy.
Oh. Okay.
Because I think they're going to
So
try to make
GFP
this
minus
point...
is no virus. GFP
Yeah.
plus is virus.
I think that they're going to
Control
try to
is
make
no
the point
injection.
that these other HVC neurons that didn't get the virus are going to help out with the recovery.
Okay.
Because the ones that got the virus are permanently...
Yeah, not able to
Right?
communicate things.
Well, the GFP plus ones in the recovered phase have a very high frequency. The GFP minus ones also have a pretty high frequency, right? They're both 200, 300% of the frequency of the control.
Yeah, what are we supposed to interpret from that?
Yeah, I mean,
Where's 5?
you know, all the things in C kind of make it look like GFP minus and GFP plus are
Yeah.
essentially the same.
Yeah.
Yeah, let's see. There's lots of HBCs.
See, because it says we
don't have to do
hypothesized that the
right.
recovery may depend on changes among other neurons that were not genetically perturbed, unmanipulated neurons
Oh, so
within
are
the
they
same
mo-
HVC.
So they're looking at the not all neurons in the HVC got the
Right, because it's lentiviral.
Yeah.
okay, so they're looking at the unaffected versus the affected.
Yeah, because then it says we performed whole cell recordings to measure the synaptic intrinsic properties of unmanipulated GFP negative neurons after LV tent. So yeah, presumably the lentivirus only gets some of them.
Okay, so the ones that don't have GFP didn't get
Oh,
lentivirus,
but the animal did.
but the animal did.
Okay.
And we see that there is increase in this MEPSC frequency for both.
Oh, so
minus GFP.
So the
I
cells
guess-
that didn't get hit are firing faster.
or what it means is it says they recruited a substantially higher level of excitatory synaptic inputs than neurons in unperturbed animals, as revealed by a much higher frequency of miniature excitatory postsynaptic currents.
Okay.
So I guess after injection, whether or not you're infected or not infected, you're receiving more.
Information.
Yeah.
So they say, thus our findings show that alongside song degradation and following recovery, the HBC network is dramatically restructured. And they're making this claim that it's restructured because there's this higher firing rate of unaffected cells.
And so then the question is, how is it restructured?
D, a schematic diagram of the neuronal organization in the model. If you say so, it just looks like a bunch of dots where every single dot is connected to
Yes,
every
I
single
think-
dot.
Yeah, I think they're saying all of them are talking to each other, and then in this the lower part, only the green ones
Are able to
are
talk to each
able
other.
to talk, or
Okay.
or actually only the red,
Only the
only
pink.
Only the pink ones are able to talk. Okay. Okay, and then E, a schematic illustrating the single cell homeostatic plasticity rule implemented.
Synaptic strength versus firing rate.
No idea what that figure is supposed to show you.
So if you have a lot of inputs and you fire to something that has a lot of things coming into it versus you have a lot of inputs and then you fire into something that isn't receiving a lot of inputs.
Then it has a lower synaptic strength.
Then it has a lower synaptic strength.
Or another way to read this is that it has a higher firing rate.
A higher firing rate. Yes. You fire more.
Because I think that's what the measurement literally is. No, because they have both the amplitude and the amplitude actually looks like it doesn't change that much. That the amplitude. So this model, the way they've drawn it, seems like it's not right about the synaptic.
No, so I think what they're essentially saying is like if you're in this super connected network,
and
you can just like fire a little bit.
They have it flipped. This is the x-axis.
What?
The change in synaptic strength, right? The synaptic strength is a thing that's being modified when they mess with the network. And so because they change the synaptic strength, right, they get a change in firing rate.
Right.
Okay. We just have control and dependent and independent variables confusing. F. Spike raster plots showing the sequential dynamics generated by a subset of the model HVC neurons before perturbation and after recovery.
Okay.
They look pretty similar.
Yeah.
Like if there's less.
Probably.
And then G, strength of synaptic input for
Unmanipulated is pink and
interneuron is blue.
Wait, are we still modeling or we're done with modeling?
I think we haven't been modeling. I think this is
Okay.
all
Well,
data.
I mean, D and E is kind of model,
Okay. Very roughly.
but that might be all they do in neuroscience. That might be what they mean by models.
No, because here they have a plot.
Okay, so maybe a real model thing later. Okay. Oof. How do I read this? H, change in firing timing. Initial firing time and cell index. What am I supposed to get from that?
Oh.
I don't know. Okay.
Change.
We found that adding this form of cell-autonomous synaptic homeostasis. Why are they using these words? What is synaptic homeostasis? Homeostasis means not changing, right?
So I think they're
Just like-
saying initially you're in a steady state. You perturb the system and it's going to come to some new steady state. Right?
New
Okay.
steady state is an oxymoron.
No, you started in one steady state. You perturb the system. You're no longer in steady state, and you're entering some new steady state. I don't understand how that's an oxymoron.
I mean, your new steady state if you keep perturbing.
I mean,
right? Like,
if you
on
talk
your
about
perturbation,
steady state like that, then like all of everything is just an infinite series of steady states.
Indeed.
Very, very short steady states.
No, indeed, you have a steady state where they're all black. No? Okay.
Yeah, I believe you. I agree with you.
And then you have another steady state when you get rid of all the green ones.
Sure. Yeah.
Okay?
Yeah.
But how else do you want me to
No,
describe
no,
this?
okay.
So that's what I'm saying, is that's what I think they mean by homeostasis.
What is the cell autonomous synaptic homeostasis? Why the cell autonomous?
I guess
Does it
because
say cell autonomous?
We found that adding this form of cell autonomous synaptic homeostasis in excitatory neurons reliably restored sequential network activity.
what figure is that in reference to?
All right, I'm reading the model. No, actually, it's hard to read the model. We sought to determine which mechanisms are consistent with restoration of the sequences, as well as with our physiological findings about synaptic strength changes. We model HVC as an excitatory inhibitory network, with HVC neurons connected to each other in a feed-forward polychronous chain, and recurrently connected to interneurons, figure 5D. The interneurons are in blue, so those are important. We then inactivated varying fractions of the HVC population, as Lynn said, to mimic the silencing of neurons caused by tent expression. To explore mechanisms that may enable recovery, we first implemented STDP between excitatory neurons, EE, which is integral to many models of sequence self-organization. What is STDP?
They defined that earlier.
Oh yeah?
Yeah.
What is it? Smooth, transversal, dumb
No,
polychains.
it was early on. It was when we were still reading. Thought. Yeah.
Oh yeah.
Spike timing dependent plasticity.
We first implemented spike timing dependent plasticity. What is that? Between excitatory neurons, which is integral to many models of sequence self-organization.
Many potential mechanisms may contribute to the recovery, including STDP.
So this must be like a well-established
Sure,
metric in
we
the
don't know what it
field.
is. Put an equation in, people. Just put it in. You can plop it in right here. We found that STDP can indeed restore sequential activity with the help of independent random inputs across the network, but that this restoration only occurs for weak perturbations. Further, this mechanism does not produce the increase in excitatory connectivity relative to the level before perturbation in neurons observed by our electrophysiological measurements. Okay, so STDP, whatever it is, alone was insufficient to restore sequential dynamics. We next considered whether cell-autonomous homeostatic mechanisms may enable recovery of the network. While homeostasis may act either on intrinsic excitability or synaptic inputs, homeostasis may act? How does homeostasis act on anything? Our recordings show that unmanipulated HVC neurons did not show changes in intrinsic excitability post-perturbation, but rather in their synaptic inputs. Thus, we added a cell-autonomous homeostatic rule into our model. I don't understand. I'm lost.
And so, like,
Can we get some equations?
why do we care? What are they trying to say? I think they're trying to convince us that
I just don't know what
the
they mean
network
by homeostasis.
can somehow recover based off.
I feel like they're using homeostasis to mean something different than I want it to mean.
Yeah, because it seems like they're trying to use it to say, like, to make some sort of comment on how many inputs are going into, like, a specific cell. Which I wouldn't necessarily consider cell autonomous.
Okay. Should we just power on and be like, we don't understand this part?
Sure.
Okay. Last figure. And then we can read the discussion. Here is some circles in serial. Looks good. Here is some circles in parallel. They don't super look like they're in parallel. They kind of look like they're in serial. Where's the caption for this?
It must be on the next page.
We're on 6.
No. Oh, here it is.
No, you
It's
got it.
on.
Before.
It's on. The
Oh, I see it. It's on the bottom of the other page. Okay. Schematic diagram showing two types of potential circuit recovery mechanisms. Left, serial is the recovery of sequential firing, requires building of feed-forward synaptic chains step by step. Right, parallel is all links in a sequence are repaired simultaneously. So it's about repair. Serial repair, parallel repair. B, normalized duration versus renditions post-perturbation. Alright, give up. I don't know what's happening anymore. We almost made it through the whole paper, and now I've got no clue. I would need to start Googling shit. I would need to read the actual paper.
Where's the
So
legend?
the title of this figure is Saltatory Recovery of Syllable Duration.
Saltatory.
Yeah, what is saltatory?
Is that a sentence?
No.
It's not?
It's not a sentence.
Recovery isn't a verb.
No, saltatory recovery. Recover is a verb. Recovering, recovery is not a verb.
Saltatory.
So a sentence would be neurons recover through, or like syllable duration recovers saltatorily.
Alright, let's read the discussion.
Okay, it does say the recovery of a full syllable duration occurred by a sudden leap from a shortened state.
I really feel like this is like neuroscience has
their completely own science.
their
they
own
refuse
language.
to work with
They
any other
just like
scientists.
they're siloed off in their
A neuroscience paper written for neuroscientists.
Yeah.
I
It
mean,
might
it
be-
is, in their defense, it is nature neuroscience, so
Yeah.
it's not meant to be for a broad audience.
Yeah, but everybody wants to write for other scientists. All right,
Okay,
round
so
table
the-
style. Megan,
Okay.
take us off.
Oh my
Discussion.
gosh. We're reading the whole discussion?
It's
It's
so
so long.
I think then we're done.
Okay,
We can do
it's
it.
so long.
We can do it.
I don't know if I can do it. I'm not excited enough.
Megan will do it in a goofy voice. We'll see that.
Here, we have observed the largely unsupervised restoration of a complex learned behavior following large-scale neuronal perturbation in a brain circuit essential for the production of the behavior.
John.
We perturbed neuronal activity using two distinct methods to probe general mechanisms of resilience in response to multiple ways in which circuits can be disrupted. We perturbed neuronal activity by increasing neuronal excitability by NACH back, and we blocked neurotransmitter release by TENT. The behavioral recovery we observed was accompanied by local synaptic reorganization that can be modeled through proposed population-level homeostatic plasticity. This model recapitulates the experimental finding that restoration of sequential firing can happen largely offline, i.e. without feedback from practice, to result in a song recovery pointing to contributions from unsupervised circuit-level reorganization. Such an unsupervised recovery of basic sequential dynamics may be necessary to scaffold feedback-dependent retuning.
So aren't we just saying, look, it heals itself? But I guess it's
Yeah.
not because it's a network, so it's kind of surprising that it's healing itself when it wasn't like literally physically damaged. It's the surprising thing that the network is healing itself.
Well, and also that the cells that get perturbed, it seems like those, from my understanding, never really recover, but
Yeah.
that there's enough synaptic plasticity that the HVC can kind of figure it out.
Yeah,
And it
knows
doesn't
what it's
need-
supposed to, like, that's, I think, the thing that confuses me is how does it know what it's supposed to be outputting?
Right, and it
Yeah,
doesn't
alright.
really seem like they need any sort of like peers helping them out to like relearn these things. It's just kind of, it's pre-programmed.
But maybe the RA is sending things back, being like, I'm not getting what I was expecting.
Yeah.
Right.
Um,
Yeah, like it would be interesting to like perturb the RA and be like, can it recover now?
yeah.
Is practice required to advance song recovery? As we showed in results, much of the timing structure of songs was restored rapidly, even without practice, and substantially restored motifs were produced immediately following prevention. The remaining timing precision and acoustics were resolved over the subsequent 2 to 3 days of practice. This is in contrast to the freely singing bird's recovery trajectory, in which song is restored after 10 to 14 days of practice. Together, these results show that a considerable number of practice renditions are not required to arrive at an equivalent state of recovery, indicating that unsupervised processes contribute to recovery. However, our findings suggest that even though the general dynamics in HVC and the backbone structure of song syllables can reemerge without feedback, practice is still essential to reestablish the reliability and consistency of singing behavior. Additional mechanisms, 2, following song degradation after lesion of NIF, a nucleus of the song system that provides input to HVC. I missed something.
It's the top of the page here.
Oh, come on. Why is there a little tiny bit of text there? Okay. Additional mechanisms to completely restore the song might include further activity-dependent refining of HVC sequential activity, regaining of muscle tone following song prevention, and a potential involvement of auditory feedback through the interior forebrain pathway to retune connections between HVC and RA.
Song recovery was accompanied by increased excitatory synaptic inputs to unmanipulated neurons within the same brain region. On the basis of these findings, we modeled sequential dynamics to test how plasticity mechanisms can support this restoration. Single-cell homeostasis has previously been proposed as a mechanism for recovery. Citation 11, if people want to understand what that means.
I just want to say, like, from watching a couple Neuron talks out, this was the bird hopping from bit to bit trying to figure out its location that we both saw. I feel like, we were talking about the neuroscience isn't like correctly talking to other scientists. They need to talk to the mathematicians about modeling. Or the physicists about modeling, but like talk to somebody about modeling. Your modeling cannot just be node-based modeling. It can't be. They've got to do something more. Okay, sorry, keep going.
Single-cell homeostasis has previously been proposed as a mechanism for recovery following song degradation after lesion of NIP, a nucleus of the song system that provides input to the HVC. Here we demonstrated through modeling that the further inclusion of population-level homeostasis is needed to account for the experimentally observed synaptic reconfiguration. This form of plasticity confers an additional mechanism for resilience as it establishes the recruitment of HVC RA neurons that did not initially participate in song dynamics, defined here as silent neurons. Okay, wait, is this true?
So, I mean, I guess what they're saying here is that if you just focus on the cells that are, it doesn't really make a lot of sense, but if you just focus on the cells that are perturbed, like, the model can't explain the recovery, but then if you focus on other cells being recruited and, like, new synaptic connections, it can. And maybe those are the silent neurons.
I thought the silent neurons, I thought the other neurons that are helping the network were already participating in the network and are just strengthening their connections.
I think
I don't know why they're calling them silent neurons.
I think that they weren't originally participating. And the reason why I say that is not based off anything in this paper, but the talk that we saw was about how if they sing a song, the same song can be sung by different sets of neurons, and it's kind of random which set of neurons decides to sing that song.
In the same bird.
In the same bird.
And he showed a brain and it was like beep, beep, boop, beep. And it was like, you know, that, but it was like different neurons, but like the same song.
Okay.
Yeah.
Providing a potential role for these neurons. Neither STDP nor single-cell homeostasis can recruit these neurons into the active dynamics. The model predicts that the set of HVC RA projection neurons active in song would shift substantially following perturbation. While HVC activity is generally stable, such population-level activity regulation could support naturally occurring representational drift, which has been observed in several brain regions wherein the qualitative nature of the sequential dynamics is preserved, but the neuron that participate in the dynamics change, potentially increasing circuit robustness. Our model thus suggests an important role for silent neurons in the maintenance and resilience of behavior after large-scale perturbation of the brain circuit. We acknowledge that population-level homeostasis and dynamic recruitment of silent neurons are not the only possible explanations for our experimental observations. For example, one alternative that may account for the increases in synaptic strength beyond the original level is that even when upregulated by standard homeostatic plasticity, newly recruited synaptic contacts could sublinearly if they accumulate in the vicinity of the existing ones. It will be valuable in the future to test these predictions using imaging or recording methods that can reliably track the activity of individual HVRA neurons in recovering animals over several weeks.
You did it. You did it.
In this work, we focused on the behavioral and electrophysiological consequences resulting from perturbations that resulted in the permanent and irreversible inactivation of neurons within HVC. In future experiments, it would be informative to explore the resilience of HVC in response to reversible manipulations with optogenetic or chemogenetic methods. In particular, it would be interesting to explore how HVC responds to temporally controlled perturbations, such as during sleep, or selectively disrupting individual symbols using optogenetic stimulation or silencing. In addition, we have focused our analysis on local changes that occur in the HVC, the same nucleus where we perturb neurons. In the future, it will be important to investigate the contributions of other song nuclei to the completion of recovery, including changes in RA. The nucleus immediately downstream from HVC and nuclei from the interior forebrain pathway, which have been previously shown to be important for some forms of adult plasticity.
That was foreshadowed by Linnea. Okay, HVC has been shown to drive song with bilateral coordination, raising the question of how the laterality of perturbations influences their impact. Recently, Ochi et al. Showed a relatively rapid... showed a relatively rapid recovery of song, about 3 days, following unilateral perturbation of inputs to HVC via NIF. It is possible that this rapid recovery was because the perturbation only affected one hemisphere. However, we find that with unilateral manipulation of the HVC circuit, the nature and severity of song degradation is comparable to bilateral manipulation, but the homeostatic cellular level changes only occur in the perturbed hemisphere. This supports the conclusion that directly perturbing HVC may require a deep reorganization of circuitry within the HVC, which we observed in our measurements of synaptic reconstructing, further supporting our model's need to introduce additional mechanisms beyond single-cell homeostasis.
The unsupervised recovery of a complex learned behavior after neuronal perturbation described here indicates that self-organized restoration of sequential dynamics may be key to enable brain circuits to support resilient behavior in the face of perturbations. These observations may have important implications toward understanding the mechanisms by which the brain ensures long-term preservation of information and the continuity of behavior and improves restoration of neurological function after disease or injury. Meh. Seems like a lot of the discussion is just things that they were forced to say.
I know,
by
I like
reviewers.
a discussion that's a little bit more speculative, where you can kind of just like go a little crazy.
I feel like it just the reviewers were like, what about? And they had to keep putting
They're
stuff
like,
in the
we'll put that in the discussion.
Yeah.
Great point.
All right. That's our first journal pub. Journal pub.
I didn't ask the classic question.
Why is this night different than all other nights?
Yeah, no. Why did you choose this paper?
Oh, but I said at the beginning, we chose this paper because me and Megan saw a talk by the last author that was terrific, and there wasn't exactly a paper online that was quite about that talk, so this one seemed pretty close.
Okay.
I actually feel like it'd be a fun field to be in, because I feel like it's like one of the most like mechanistic systems to actually study like what the brain is doing. Like where you can actually
It's so
like
cool.
perturb something and see a different behavior.
This HVC bird Finch model is so fucking cool.
Should we listen to that bird song?
Yeah.
Maybe we can play it, yeah. Yeah, I was blown away that you could get something so simple and so tractable and like actually like make biological progress. That's pretty cool.
I wonder why I'm so unexcited about neuroscience.
You didn't see the guy talk.
Well, but I saw these great talks at HHMI, but I feel like every other talk at HHMI, or every 2 out of 3 talks are neuroscience talks. The best ones were the bird ones.
Do you feel like it's not mechanistic enough?
I just like it.
Or do you think it's very niche?
Well,
you
it's
feel
just
like it has no physics.
It's like what is it telling me, and like what are you measuring, and
Right,
I feel
like
like
the bird songs were like they were beautiful talks, the ones that I saw at the last conference.
I feel like they're right on the cusp
awesome.
of having good enough measurements that you can start to like see where the physics can be. It's right on the cusp of like you can almost see a mean field theory being applicable and testable here, right? And so it's like it's exciting to me because they're like a year or 2 away from like this being tractable to physicists. And like when we saw the guys talk, I was like, oh, they're there, right? Because the guys talk was like a year or 2 in the future.
It's also like I feel like neuro is like separate enough that like Princeton has its own neuroscience institute. That it's like they're kind of doing their own thing.
Their own thing, for
and
sure.
I don't think they really like maybe they don't really identify as like molecular biologists or physicists.
I went to a Princeton neuro talk like last week as it was on bird neuroscience again. There wasn't a single person in the room I'd ever seen before. I've been here for 4 years now. I've never seen a single person in that room before. So it's very siloed. Yeah, and it's presented in a very different way, right? Like tiny figures, not super well labeled. The sentences aren't like the figure sentences aren't sentences.
Yeah. And I think I just find this network stuff just intellectually not beautiful, which I think is just a failing on my own part.
If it was presented as more like, do you find when you're doing like morphogenesis, these network models of the tissue for like the force network stuff that like Sebastian does, do you find those uninteresting? Yes. Unwilling to say it, but
I mean, I'm not sure. I haven't given them a lot of thought.
Yeah.
I mean, they also just kind of lost me on the last 2 figures.
Yeah.
So I was like pretty on board for the first like 4.
Yeah, I don't know what we're supposed to get out of those. All right, good. What would you change about the way we do this? Like, do we think we could sit at a pub?
I don't know about that.
Yeah.
I mean, I think
I
you
just- I
could, but
really
there's going to
can't-
be
I
ambient
can't.
noise.
I can't process when I'm talking.
Yeah.
I
Yeah,
miss
yeah.
that whole paragraph. I can't focus it up.
Mm-hmm.
If there's movement, I can't.
We could pre-read it. It just seems like a...
I was trying to figure out if you could read without looking at the figures. Like, figures allowed.
No
Then-
figures allowed. Oh, it's the
Because that was
opposite.
actually
That was-
my
I mean-
suggestion was to just do figures and don't do text.
To be fair to them,
but
we didn't read 50%.
Right,
So
absolutely.
to understand the stuff at the end, like, it
So we-
probably was
In the text.
introduced.
Yeah. We could do it the other way around with no figures, text. We could do it all.
'Cause then
figures
you know-
and
I
text.
don't love reading out
I
loud.
don't love reading out loud.
I mean, as a format, I don't know. We're like,
No one's gonna be able to pay attention, like understand it. Right?
Yeah.
is it helpful if one person knows what's going
I was
on?
wondering this. I had a similar.
I
thing.
hate doing
Yeah.
But then
that.
Well,
you're the, like,
yeah, I was
you're
gonna
the journal
say.
club.
But then
Yeah.
is it worse to do that, or is it worse to have everyone read the paper before?
I think
I think it's better to have everyone read the paper. The one person reading the paper is the worst format.
But like,
Okay.
it's gotta be like you.
Or not even one person reading the paper, but one person being like, I know what's going on.
One person being in charge is the worst format for me. Because it's just like basically that one person who has no fucking
Yeah.
clue gives a shitty lesson to everybody else, and there's no like deep conversation.
That's
Right?
fair.
Either the conversation should be super deep because we've all read it, or super shallow because none of us have read it.
Right.
yeah. I mean, maybe it would work for a short paper. Like I
To
guess
read it.
to read it. We read this one paper that Lauren sent us on the plane out loud to each other.
We got 3 paragraphs into that
Okay.
paper.
I thought that was good.
We got 3 paragraphs. I'm not like being
Yeah, yeah.
hyperbolic. We never got anywhere into the
Yeah.
paper. That was good, This was good with the first 3 paragraphs.
Yeah, I don't know.
Yeah, we'll have to think about it. I guess we could say to anyone who looks at this, like, recommend a format of how to
That
do
would
journal
be
pub.
a pretty boring podcast to like
To just read it.
have a minute of listening to them just read a paper.
Yeah, we have this problem for the actual podcast as well, as we try to start with like the little, not the abstract, but like the little importance clause that they put on published papers,
Mhm.
and you're like starting with the most boring part of the whole thing.
Mhm.
Okay, I'm gonna stop the recording.
Yeah, I think we
Oh, we should say bye. Bye. You guys have to say something. It's mostly audio.
Bye.